Good morning, everyone. Welcome. It is my absolute immense pleasure to welcome Dr. Carmen Canavier for this talk. Carmen and I have been collaborating for a long time, since I was a PhD student. So I'm very happy to be able to invite her here. Carmen got her undergraduate degree at Vanderbilt in chemical engineering. And then she actually worked in industry. She was the first female engineer at a shell drilling platform in the 1980s. Then she quit the cushy industry job and went to the dredges of academia. She got her PhD in electrical and computer engineering at Rice, and then did a postdoc in neurobiology at UT Houston. And one of the great things that Carmen and her lab are, I mean, for certain in my view, but probably I shouldn't be so certain. They are the world's experts in the modeling of dopamine neurons and understanding the physiology of like how dopamine neurons function from a computational and quantitative point of view. And that's what she'll be talking to us today about. So without further ado, Carmen, please.
Thank you for that kind introduction. I guess you can hear me okay? All right. Anyway, so as Cowie said, I'm going to be talking about computational modeling of subpopulations of midbrain dopamine neurons. These neurons are located in the substantia nigra pars compacta and the VTA. And this work is funded. In fact, Cowie and I are co-PIs on the NIDA grant, a dynamic diversity of dopamine neurons.
Last time we could use diversity in the grant.
Okay, this works before. How do I switch slides?
You can just press
Yeah, this doesn't work anymore.
Let's see. Like, it does not work. That's crazy.
There's always a technical glitch.
There you go.
Okay.
I just clicked.
Okay. All right. So anyway, this is a nod to my collaborators, including Cowie here at UAB, Chris Knowlton in the audience, and Ria DeHal in the audience, and Jochen Roper and Marlej Janki and let's see, and Strahi Sojanovic did most of the work in this that I'm going to be presenting. So this is called at least in Jochen's lab, it's called the obligatory Schultz slide. It's when he was recording from monkeys and gave them an unexpected reward, a drop of juice. And these are actually, spike time histograms from repeated trials in the same neuron. And you can see that after the reward is received, the dopamine neurons have a burst. So this is a very widely known, a famous finding. And what was interesting also is then once he gave the monkeys a conditioned stimulus of some kind, a tone or a light, then they ceased, after they're very overtrained, they cease firing to the reward, which is the US, and they start firing only to the CS, which is a predictor of the reward. And then in these highly overtrained animals, you get this, you now present the CS and they expect the US, but they don't get it. Then there's a pause in the firing.
I'm just going to try
Okay.
to minimize this. You can.
Okay. Oh, I'm okay. It doesn't bother me. Maybe it bothers the audience. But in any case, but originally Schultz thought that the dopamine neurons were a homogeneous population, and they all kind of did the same thing. But Jochen Roper, who was Cowie's mentor, was a leader in publishing on the diversity of dopamine neurons. They're not all the same. There are, and okay, now I can't switch, Cowie.
Seems to be.
You made it worse, Carmen.
Yeah, it seems to be. Now I can.
Okay, he's got the magic touch. Yeah. All right, but anyway, so there are subpopulations, and according to Stefan Lamel and Jochen that started this work, the subpopulations are based not so much on location but on projection target, and they have different responses. And so the original population that was first discovered, they're conventional in our terminology. They're more lateral. They were identified first, but then Stefan Lamel in 2008 identified, and when he was in Jochen's lab, identified another population that we call atypical. And there are several differences. One of them is that the conventional ones, if you drive them with excitation, this is a triangular ramp. We'll talk about it a little more, but they don't fire very fast before they go into depolarization block, whereas the atypical ones, they can fire faster before they go into depolarization block. There's a differential regularity of pacemaking. And so these conventional ones, we define that largely by the compressed dynamic range, whereas atypical have extended. But there's some also other differences that we'll talk about, that the conventional ones have more regular, their pacemaking is more regular. They have different responses to hyperpolarization, which is shown at the bottom. So there's most of them, dopamine neurons, have some sort of a post-inhibitory ramp response that amplifies the inhibition, for example. But there's a subpopulation, and that's only a subgroup of the conventional ones that responds to inhibition with a rebound burst. And that's what you see. You see the rebound burst here. You see a ramp here. And so I'm not sure how many of these differences I'm going to get to, but we'll go with differences in response to excitation, different regularity of pacemaking, different responses to inhibition. And finally, there's a special type of burst that Kaneko Otomo in Johan's lab identified that's a plateau burst. And we find that we predict that those are going to be more likely in the atypical. And here's a summary. Yeah, okay. Well, it's okay. Here's a summary of differences, electrophysiologically speaking. On the left-hand side, there's the data from Stefan Lemmel's paper, and the difference in frequency. That's not something that I want to emphasize. But the difference is that they can fire faster. But here, what we see is you've got a very pronounced AHP after hyperpolarization potential in the conventional compressed range. You have a narrow action potential that's taller, so they're shorter and broader in the atypical population, and then this different shape of the action potential is reflected here in the phase plane plot. So unfortunately, yeah, okay. So you can see dV/dt is that's the y-axis, voltage is the x-axis for all these plots. These are all phase plots. So dV/dt versus voltage, and you can see that it's kind of squished in the atypical neurons, which means they have weaker spiking currents. So the atypical has a shallower AHP due to less SK current. They have weaker spiking currents reflected in their shorter, wider action potentials. And then the ramp responses reflect that there's a slower recovery, or slower inactivation of KV4 in these channels. So upon hyperpolarization, you recruit KV4 A-type channel, and then as it stays on, you don't fire, but as it inactivates, then you start firing again, and so it just takes longer in the atypical. And one final difference is if you look at this here, it's particularly on the right-hand side is the model, on the left-hand side is data. And if you look in particular at a response to a hyperpolarization, you get a sag current. And that's due to IH. And that used to be used to identify dopamine neurons. And there was, in fact, a paper from Tony Grace's lab that said, are you or aren't you? And that was when they were talking about, so yeah, maybe, now just the presence of a sag current is not enough to identify a dopamine neuron, because this atypical population doesn't have it. And the way in the Lemel study, they expressed DAT, TH, and VMAT to prove that it was a dopamine cell. So, all right. And this is again from the Lemel paper, but it's just to illustrate that they're not grouped by where they are. So there's the parabrachial pigmented nucleus, the paranigral nucleus, and the intervesicular nucleus. And of those, those neurons, they can project to, they can be mesocortical, they can be mesoamygdaloid, and they can be mesolimbic core. And it doesn't correlate exactly with their location. Their projection target doesn't correlate with the location. The conventional ones pretty much all project to the dorsal striatum or to the lateral shell of the nucleus accumbens. So medial shell and core of the accumbens projecting are atypical, whereas lateral shell projecting are grouped with the conventional ones that were project to the dorsal striatum. And this is a paper that Cowie's name is on here. It's just illustrating. Now, I was showing you VTA neurons. This is now SNC neurons. And so they also have a variety of projection targets. And we're going to talk later about a special group that projects to the DLS. So the DL, they have, the same, you can project to the DLS, DMS, lateral shell of the nucleus accumbens, medial shell of the nucleus accumbens. But we're going to focus, like I said, we have these two groups, atypical and conventional, but then within the conventional, there's a subgroup that projects to the DLS that has a special response to inhibition that we're going to hopefully get to. right, so this is again a figure from Stefan's paper. I put the triangular ramp in there. That's not in the paper, but he's applying a triangular ramp of current, depolarizing current, and he's showing now he's identified the projection targets. I'm not an experimentalist, but I think this involves putting a virus in the projection target, and it goes back to the dopamine neurons. All right, so in any case, the mesocortical, mesoamygdaloid, which I didn't mention before, mesolimbic core, mesolimbic shell, these all have a limited dynamic range. They can fire, I'm sorry, they do not have a limited dynamic range as much as the others. They can fire at 20 hertz before they go into depolarization block. So the reason they stop firing is they're in depolarization block, which means that their sodium channels are all inactivated, and now you can't recruit enough sodium to produce a spike. But the conventional population projecting to the lateral shell and the dorsal striatum, they can only fire at maybe 10 hertz before they go into depolarization block. And they also go into depolarization block a little differently, that the spikes fail more abruptly. And we talk about that in the paper, but I don't have time to go into that in this talk. All right, so now we get to the computational modeling methods. And for most of the talk, I'm going to be talking about single compartment models. And this is where my electrical engineering training comes in. We modeled the neural membrane as extracellular fluid, intracellular fluid, as the membrane as a capacitance that's in parallel with a bunch of nonlinear conductances. And conductances, as you know, is the biological term for 1 over the resistance. And then each neuron, each current has a battery that's provided by the concentration gradient that's set up by pumps in these neurons. And so we do an electrical equivalent circuit of the membrane. And then we also do a material balance on calcium ions. And in this talk, we're mostly going to be, there's bulk cytosol, but we're mostly going to be concerned with a microdomain around the SK channel. All right. And just since this is, there are engineers in the audience, I put a equation slide I don't usually include in my biology talks, and I hope no one's offended by that. But in any case, so for the membrane, it's a differential equation. It's just CMdV/dt is the sum of the currents. And this is from the definition of capacitance and from Kirchhoff's current law. Then for the individual currents in a Hodgkin-Huxley type model, it's just Ohm's law with a nonlinear resistor. So we have a battery, we have a maximal conductance, and then you have gating currents. So here M represents activation gates that open the channel and an inactivation gate that inactivates the channel so that it doesn't pass current. And then and this is just basic Hodgkin-Huxley, for the activation gates, they all have the form that the derivative is just a relaxation to the steady state value as a function, a first order differential equation, as a function of their steady state value at that particular membrane potential. And at that particular membrane potential, they also have a time constant, which they relax to their steady state value. And then the electrical driving force, the way you get the batteries, that's from the Nernst potential, where the electrical driving force equals chemical driving force. And then the material balance is just the calcium currents coming in minus the ones going out, and divided by a conversion between charge and mass. So the first thing I'm going to focus on, and I need to go back to the equation. So the first thing I'm going to focus on is the voltage-gated sodium channel. That's the one that's responsible for the upstroke of the action potential. And that's since depolarization block involves that channel, obviously we try to model the differences in the subpopulations. We're going to focus on the sodium channel. And what happens if you do a regular Hodgkin-Huxley model here with the M cubed H formulation that they got the Nobel Prize for in medicine and physiology in 1952, I think it was. What happens is if you apply a triangular ramp, a current ramp, so this is atypical, the data goes into depolarization block, it stays in depolarization block. With just fast sodium inactivation, you go into depolarization block, but you come out of it as you hyperpolarize. So we had to do something different. And what we did different was, instead of going with the Hodgkin-Huxley model, which is shown schematically on the left, where you have 4 independent non-interacting gates, 3 to open the channel and 1 that causes it to inactivate. So all 4 gates have to be in a conducting state for current to pass. And again, I repeat the equation, the Hodgkin-Huxley type equation for sodium. But we had to go, in order to capture the difference between the populations, we had to go to a Markov model. And a Markov model just has transitions between states. And if you're in one state, you can't be in another. So they're, by definition, they're not independent. They do depend on each other. And then the current is then only the open state passes current. So the fractional occupancy in the open state is what gives you the current. Okay. And then as you write differential equations for the gating variables, you also write differential equations for the transitions between the Markov states. And these transitions are, in general, voltage dependent. So the rate in minus the rate out gives you the accumulation of any given state. And with the caveat that the fractional total occupancy at all states must sum to 1. So you need to ensure that. All right, so there's an interesting molecule that I've recently become aware of, and that's called fibroblast growth factor FGF12. And one of its functions is it is a substrate for a long-term inactivated state in voltage-gated sodium channels. It also has other functions that I'm not familiar with. There's a receptor, there's a binding to signaling proteins, that has actions in the nucleus and regulation of cytoskeleton. But so we added to we made a Markov model and added an activation state that we called I1. It's like the Hodgkin-Huxley state because you go into it fast and you come out of it fast. But what we changed is we added an I2, which you go into it fast, but you come out of it slowly. That's why it's called long-term inactivation. And when we did that, so at the end, I've repeated Stefan Lammel's data on the compressed dynamic range versus the extended dynamic range. And this is Chris's model, showing that the neuron, and you're given it a triangular ramp of current, and that as you give it this ramp, it does go into depolarization block and it stays there and it fails abruptly. And then the atypical model, and the only significant, there's some tweaks. We take into account that they have smaller spiking currents, that they have a weaker SK channel, a weaker H channel, and that the inactivation time constant for KB4 is different. But none of those changes made a qualitative difference. So the only thing that made a qualitative difference was adding this slow inactivation and making the conventional population with more susceptible to go into this long-term inactivated rate. So the rate here, the rate from I1 to I2 in this model is larger. But the important thing is just that they are more likely to go into that state than the other ones. And what we show here is occupancy in the various Markov models. And the red means that you're in one of the two inactivated states, which means you're not available for an action potential. And once you get too many of those in that state, then you can't fire action potentials anymore, and you're in depolarization block. So that was successful. But what's interesting is, this is one of the main differences between, say, the conventional and the atypical neurons. And this is an unpublished figure from Jochen Roper's lab. And interestingly enough, the one change in this mouse model of schizophrenia in the dopamine neurons projecting to the dorsomedial striatum is a downregulation of FGF12. And then there's also an upregulation of NMDA, GRIN2C that would upregulate the NMDA conductance. But and I'm not going to go through this. I'm not going to go through all the details in this figure, but it's just basically that these are the neurons. So it's a UMAP cluster of all the dopamine neurons, and then on the right, it's only the ones that project to the dorsomedial striatum. And then there's there's the wild type, and there's this model of schizophrenia, mouse model of schizophrenia. And so there's no difference in all DA neurons if you look at them all together. But if you look just at the DMS projecting ones, then this protein, this accessory protein, is downregulated. And so with that, we haven't done the modeling yet, but it's obvious that that's going to turn a neuron that projects to the DMS, the cognitive circuit. It's going to turn it into more of an, it's going to switch the phenotype. Because if you don't have as much entry into this inactivated state, you're going to act more like a conventional neuron than like an atypical one. So this may be important to schizophrenia. We were very interested to find out that the one thing we identified as being the major difference in the electrophysiological phenotypes is indeed regulated in this mouse model of schizophrenia. So So that's all I'm going to say about the differences in response to excitation and going to depolarization block. But next I was going to, and this next topic is maybe the only one that I get through actually, because it's a pretty complicated one. And it's about pacemaking regularity. And so we're using, basically the same model, similar model, single compartment, but now we're going to focus, and I didn't mention, this little dash line, that just means that we model the the calcium near the SK channel as microdomain. So there's in the models we can have different nanodomains, different microdomains, different places for calcium accumulation. But it's important because the SK current is going to be important, very important in the regularity of pacemaking. And remember that the biggest difference that Stefan Lamel found between atypical and conventional was the depth of the AHP. And the depth of the AHP is largely controlled by the SK current, the calcium activated SK potassium channel. But then IA is also going to be very important. The KV4, the A-type transient outward potassium current is also going to be very important in this phenomenon. So this is actually unpublished. It's in bioRx. It's not out yet. But the top is data from Jochen Roper's lab on the atypical neurons projecting to the medial shell of the nucleus accumbens, and then we've got some conventional ones projecting to the DMS and DLS. And this is data, and these are phase plane plots. And again, you see that the atypicals are squished because the currents are smaller. But what you also see, and apparently everybody that records from these neurons knows that the atypical neurons are less regular. Their pacemaking is less regular than those of the conventional neurons. The conventional neurons are little regular pacemaking clocks. And this is data from Jochen's lab showing that the CV of firing during pacemaking, and I didn't mention, and I probably should have, the fact that dopamine neurons in vitro are spontaneous pacemakers. I just assumed everyone would know that, but that's not a generally known fact. Yes. So if they put them in it, and Cowie records for them, maybe other people could, but they don't need, and Jim Surmeyer had a paper once that said, who needs excitatory synapses anyway? Because all the neurons in the basal ganglia are pacemakers, or most of them. So dopamine neurons are pacemakers, and like I said, you note that in the conventional population, there's a ramp. There's a very regular ramp between spikes, whereas not so you don't, it's dependent on recovery from that AHP, and you don't see it in the atypical neurons. And again, this in the right-hand side is just emphasizing that the atypical ones are projecting to the medial shell of the nucleus accumbens, whereas the typical ones are projecting to the dorsal striatum. All right. And this is some data from Birgit Liss's lab. And she just shows that in the convention, this is another difference between the conventional and atypicals. In these conventional neurons, there's this oscillation in calcium that accompanies pacemaking. So why do we care about pacemaking? The fact that dopamine neurons are pacemakers in vitro. Well, for one thing, it gives them the ability, I think, to signal both in a positive and a negative direction. If they weren't pacemakers, then they couldn't have a pause to signal a worse than expected outcome. Whereas if their firing goes up, it's a better than expected. So it's a bidirectional signaling. But both Jim Tepper and, like I said, Conoco and Roper's lab and Carlos Palladini's lab have shown that pacemaking can persist, at least in anesthetized mice. And these are putative lateral shell projecting conventional VTA neurons. Since these are definitely VTA neurons, and we think that they are conventional because of the regular pacemaking that they're exhibiting. And so these, like I said, I'm just trying to make the case that maybe pacemaking could be important. And then Charlie Wilson has a paper, Mr. Basal Ganglia, where he talks about how pacemakers integrate their synaptic inputs differently than quiescent neurons. And specifically, if you're firing already, it's not like you're inserting and removing spikes necessarily. You might just be changing the timing and making it more irregular. And there's a different integration window for pacemaker versus a quiescent neuron. And for pacemaker, it matters when the input gets there. There's a whole thing on phase response theory that says. So anyway, that's just background. Now I'm going to try and segue into nonlinear dynamics. And I showed you a phase plane picture before that had dB/dt on one axis and voltage on the other. This has voltage as one variable. And what we see is a pacemaker on the right-hand side, that's your voltage versus time. That's your time series. If there's, in many cases, there's a rate limiting slow variable that constrains the dynamics. And so then you have a different representation where as you're traversing the action potential, you're moving around a limit cycle. So this is what's called, it's called a nonlinear oscillation is a limit cycle. And in many cases, I think that a rate limiting slow variable can constrain the dynamics. So you live in this is it's called a phase space. And I'm going to be relying heavily on the concept of a phase space. And for the first example that I'm going to show, I'm going to look at something very simple. So I used to collaborate with Paul Shepard. And when he puts on TTX and sometimes a little TTA on these conventional lateral dopamine neurons, they're pacemakers, they're firing regularly. But then if you block spikes, there's still a persistent underlying oscillation. And there's also that corresponds to the calcium oscillation that Birgit showed in the original slide, because this is due to calcium coming in and the SK current turning on and hyperpolarizing it, and then calcium coming in and the same, in a cyclic fashion. And that's a simpler example than if you have spiking. So I thought I would just start with that to explain the premise. And on the right hand, so this is a simulation. It's a little spikier than the real slow oscillation, but this is a simulation of the slow oscillatory potential. It's subthreshold. There's no action potentials. And we do have a slow variable that controls the dynamics. And Charlie Wilson's published on this. Jim Surmeyer's published on it. And that's calcium. And so what I'm showing here is a nullcline, a voltage nullcline. This is a common, it's a nonlinear dynamics tool that you have a phase plane. And then on a nullcline, that means that for that particular variable, which is voltage, the derivative, time derivative is 0. So for voltage, that means the inward and outward currents are exactly balanced at the pairs of values of membrane potential and calcium on this curve. And you'll notice it has a Z shape. And the Z shape is because this middle branch comes from what's called a negative slope conductance region. And that is simply positive feedback. So it's positive feedback, meaning that, so for example, you have an inward current and you're on this middle branch and you depolarize, and dVdt is 0. So you think you could stay there, but you can't, because if you depolarize a little bit, that opens more inward current channels. And so you move away. It's positive feedback. And on the same thing, if you hyperpolarize a little bit, then you close those inward channels, which hyperpolarizes even more. So you get pushed away. You can't stay on that middle branch. So you have to and this goes back to what we were talking about, stability. I mean, if you do a linear stability analysis of a fixed point, which is, I was talking about the black curve, which is Z. The green curve is the nullcline for calcium. So there, the derivative of calcium is 0. So calcium influx is balanced by calcium efflux. So the only place that the system has a fixed point or an equilibrium point and it could stay is at the intersection of the nullclines. But if you did a linear stability analysis of that fixed point, it would have it would have positive eigenvalues in a real system, and so it would be the voltage would grow and it would be pushed away. The perturbation would grow, and you'd be pushed away from this fixed point. But you can, so you would be, this is a nonlinear, it's called a relaxation oscillator. You'd be forced to traverse, and that's what's shown in purple is an ideal relaxation oscillator where you move along the stable branches of the voltage nullcline, the upper branch and the lower branch. And then at the end, when a stable branch ends and turns into an unstable branch, that's called a knee, and you've got to jump. You've got to make a jump. And the assumption, this is called fast slow analysis. And the assumption is that this slow variable, in this case calcium, changes very slowly compared to voltage, which changes fast. So changes in calcium are in the horizontal direction, changes in voltage are in the vertical direction. So you're going to jump in this direction, and then the evolution of the slow variable is going to cause your trajectory to move slowly. That's highly idealized. That's when you have perfect separation between the time scales. The real trajectory is shown in red. So, I mean, the nullcline analysis can guide you to think about how the trajectory is going to evolve in the phase space, but it's an approximation. All right, so I want to now move to a real case where you've got spikes. And now you can't really represent that in the two-dimensional system, because what happened, here's the Z-shaped nullcline again, and the bottom branch is going to be quiescent The middle branch, as we said, is unstable. You can't stay there, so you have to jump. But here in this case, the top branch is a spiking branch. So you need a dimension that I'm not showing. We have it in some of our papers, but you need a dimension that's perpendicular to the paper so you can spike. But you can it's good enough just to say lower branch is quiescent, upper branch is has spikes on it. And now I'm going to get to the issue of regularity and what phase plane and null-cline analysis has to do with regularity of pacemaking. And on the left-hand side here, this is from Zaid Khalek's paper when he was in Bruce Bean's lab on, he went into the VTA and he picked the most regularly firing neurons. So we now know that from Stefan's work that those are probably lateral shell projecting conventional VTA dopamine neurons. And he plotted, and you can see that there's an action potential, and then there's an after hyperpolarization, and then there's a kind of a linear ramp, and then there's a spike. So there's kind of three phases. And he showed that he then estimated the current by multiplying capacitance times dV/dt. And so the current, this is picoamps. So and it's truncated, obviously. The current's big during spikes. But in between spikes, the current's like on the order of a picoamp. There's a tiny little current there, and that's how you get slow pacemaking. And then in order to explain this, I need to talk about the A-type potassium current, Kv, in this case, Kv 4.3, transient outward potassium. And on the right-hand side here is Chris's simulation of this regular pacemaking with the three phases. So spike is in red, AHP is in purple, the ramp is in, I'm not sure if that's orange or yellow. My color of vision's not that good. We'll call it orange. All right. But anyways, so the important current that we're going to focus on is this transient outward current. And so it has a gating variable for inactivation. We're going to call that Q. It has a gating variable for activation. We're going to call it P. It doesn't matter. It's like, it's just, just arbitrary. But so the Q is very slow compared to, we're going to say activation's fast. You just set it to a steady state variable as a function of membrane potential. And in fact, you set all the other variables to their steady state variable as a function of membrane potential. And that's how you get this representation in just two dimensions. Because on this voltage null climb, which once again has a Z shape, all the gating variables are at their they're fast. So everything is fast. It collapses to the voltage null climb. Everything is at their steady state with respect to voltage, but not the inactivation of the A-type current, because that can have a time constant on the order of 50 to a couple of hundred milliseconds. So therefore, that is now I'm identifying a slow variable as being the inactivation of the A-type channel. And this dotted line here is simply it's this sigmoid that shows the voltage dependence of an activation, but now I've flipped it. It's on a different axis. So this is just this is actually the null climb for the inactivation, because it's a steady state value where the derivative of the activation variable is 0. So that is the Q null climb, should say. And during a spike, this is you hyperpolarize after the spike due to SK channels. So the slow variable here is not plotted. It's the slow variable here is actually calcium. Calcium is the operative value, and the SK channel is keeping you hyperpolarized. While you're hyperpolarized, notice you're moving to the right. As you move to the right, that means you are increasing availability of the A-type channel, and so you're removing inactivation from it. The number is getting bigger, so it's more available. So this is key. During the AHP, you've now moved the trajectory so that it reaches this narrow channel between two null climbs. By definition, null climbs dV/dt is equal to 0. So nothing can change very fast here. And the trajectory is actually confined to this region between the null climbs where the dV/dt's, the rates of change with respect to time are very small. And what drives the movement is as now we're at more hyperpolarized, we're at more depolarized potentials. And what's driving the leftward movement is the inactivation of the A-type channel. So that is the slow variable that's controlling the dynamics. and you're on this lower quiescent branch. And once you reach the knee, then you're allowed to spike. And then once you've spiked, the SK channel forces this big AHP, which drops you down into the regime. So in a null-climb plane, the plane is separated into two halves. So above the null-climb, in this case, the inactivation is removed, and below the null-climb, below the null-climb is removed, meaning the number gets bigger. Above it, then you inactivate. So that's why the trajectory switches directions here at the steady state curve for A-type channel inactivation. All right, so what is the consequence of having this narrow channel where things are moving very slowly? And so this is a complicated slide, but these are Chris's simulations. Now we've taken both the atypical model on the left-hand side and the conventional model on the right-hand side. So the conventional model is what I just showed you. There's this Z-type null-climb and there's a channel. And now Chris is giving it a bunch of noise, simulating okay. At one time we were giving it the balance state, which was synaptic inputs, but we moved to channel noise to be more like the situation in a slice because we're comparing this data from a slice preparation. But nonetheless, whatever type of noise you give it, remember that relaxation in the vertical direction is very fast. So if you perturb it, if you get a little perturbation, then you're basically not going to change the trajectory very much, and you still have this slow, regular pacemaking. But on the other hand, in the atypical, you do not have a deep AHP. You don't have much SK, so you never uncover the A-type current, and you never move to the right. You never get in this channel, and therefore any perturbation can cause a spike or stop a spike. And then if you block the SK channel, then you lose the regularity you didn't have any to begin with in the control, so blocking Apperman doesn't do much. I mean, blocking the SK with Apperman. And when I say Apperman, this really isn't Apperman. It's the SK channel being set to 0. This is a model. So, and here's the SK channel being set to 0. Again, you deregularize the firing. So, yeah, and this is just a reminder of this is the noisy simulation, and this is the noiseless one. But it just provides I think it might be a fundamental thing that if you have a slow variable that controls the trajectory in between spikes, that can be a way to regularize pacemaking, and in a general sense, and it also highlights one of the differences in populations. But this slide was put in for the benefit of electrophysiologists. I don't know if I have any electrophysiologists in the audience, but Chris sampled he did what's called an instantaneous IV curve. So, you're at steady state at this point, and then you do an IV curve from there, or you take the conditions at this point and as your initial conditions, and then you do steps to different membrane potentials, and you record the current that is required to hold it at those steps. And in this case, these instantaneous IV curves are very steep. The slope of the instantaneous IV curve is the conductance that restores you back to that original point. So you have a very steep and a high restorative force in this case because you've got KV4 that's active, and if you perturb it, it's going to pull you back. And then the atypical, on the other hand, these instantaneous IV curves are very flat, so there's not any restorative force. Anyway, just an alternate way of explanation in case there were electrophysiologists in the audience. All right, so I'm getting close to the end. Conventional and lateral shell projecting neurons. Remember, those are lateral shell. All right, so we have two instances of conventional shells. Let's see, yeah. So they all have what I'm showing is lateral, DMS, and DLS. So no atypicals here. We're all conventional compressed dynamic range neurons that were of the type that were identified first. But nonetheless, there's a difference in that so most of the conventionals do exhibit a ramp response. The ramp response is notably shorter than the ones you would see in the atypicals that project to the cortex, the amygdala, and the core and medial shell. of the accumbens, but it's still a ramp. However, there's this anomaly, the neurons that project to the DLS, if you hyperpolarize them, they respond with a burst. And this is data, and this is published. So you can see so we think that maybe, and Cowie and I have been working on this, maybe that's a global signal that synchronizes the population for movement initiation, for example, in the DLS. But this is, like I said, published data from Strahi and Jochen's lab showing that the rebound delay, latency to the first spike is much shorter in the DLS compared to the DMS and the lateral shell projecting. And then the rebound frequency, and this is, successive ISIs after the hyperpolarization, the rebound frequency is much higher in the DLS. So in all the other models that I showed you, we used a single compartment model. In this case, we went to a multi-compartment model that has separate compartments for axon initial segment and axon bearing dendrite and non-axon bearing dendrite and soma. And it's not actually, it was important for some of the phenomena that we tried to capture. And it may in fact be important for this phenomenon because the special population that project to the DLS, those have a ventral dendrite into the part of the SNR, the pars reticulata, and that receives GABA B inputs, and that's the only known GABA B inputs into these cells. And so Rebecca Evans has a paper out saying that these cells are, and she didn't actually know the projection, but they were cells in the substantia nigra that were specialized for rebound. Because the GABA B obviously is going to have a much deeper, it has a reversal potential of potassium as opposed to GABA A, which has a reversal potential of chloride. So it's going to have a more effective hyperpolarization and be more effective at evoking rebound. So these spatial nuances could be important. They're not important for the phenomenon that I show. And this is, again, Chris's work. And so there's a spatial distribution. T channels are located mostly in the dendrites. KB4, he put a gradient there. They're known to be, well, there's known to be more H-type current in the axon-bearing dendrite from Vince Sutan's lab. And of course, we're going to have more sodium channels in the AIS because we want the action potential to always initiate in the AIS. And he showed, so we're going to focus on the T-type current and the A-type potassium current as being the major determinants on why you get a rebound burst in the DLS projecting and not other populations. And so here the CAV3 that mediates the T channel conductance is larger in the DLS population. And there's a master's thesis from Jochen's lab that confirms a medial to lateral gradient in this conductance. And the ones that project to the DLS are generally more lateral. All right, and then there's another major difference is, again, we've talked about the time constant of inactivation of the A-type current, and it is longer. It's shorter. I guess what's relevant, it's shorter in the DLS projecting. So the A-type current is not going to be as effective in enforcing a post-inhibitory ramp in the DLS as it is in the other subpopulations. And so this is, I showed you the experimental data from Strahi's work. This is now Chris's simulations of the DLS, and you see the rebound burst where in the DLS only, whereas you see ramps in the DMS and the, that's shorthand, in the DA neurons projecting to the DMS. So this is DA neurons projecting DLS, dopamine neurons projecting DMS, dopamine neurons projecting to the lateral shell. And again, the rebound burst only occurs here. And interestingly enough, data has shown that HCN, that the H current expression is not that different between these populations. So that's not mediating the difference in rebound, but it is instead the T-type channel. See, you get more T current here, more CAV3 in the DLS compared to DMS. And this KB4, you get more of it activated because of this burst, but it also, so the H-type current is turning off faster. You can see it's turning off faster in the DLS. And so this is all ex vivo work. So this is all in slice that you have these ramps. So Chris did some simulations to see if this had any relevance to the situation in vivo. And so he simulated a spontaneously active balance state in which the dopamine neurons are continually being barraged by IPSBs and EPSBs, synaptic potentials, excitatory and inhibitory synaptic potentials. And this, correct me if I'm wrong, Chris, but I believe this is actually a population. Yeah, it's a population. It's not a single cell roster like the ones that Schultz does, but it's actually a roster across. Chris generated a bunch of different parameter sets that were consistent with these core projecting.
Different cells in different balance states.
Right. Okay. But so anyway, they're consistent with the properties, but it's now a population. And when he applies there's a right here, he applies a burst of IPSPs. And so, of course, it's quiet during the IPSPs, but it's also there's a pause after the IPSPs are done. So this pause that amplifies inhibition, we predict that it would and on the bottom here, it's just a histogram of mean frequencies, just another way to quantitatively show that you do have a pause that exceeds the duration of the IPSV. So we predict that it would still be relevant in vivo. And the next point, I think, well, I'm debating on whether I have enough time to go into it. It's a short point, so I will. All right, it's short. So again, now we're going to go back to the single compartment models. Now the important currents are again the SK potassium current and the M-type Kv7 current. And remember, a Kv7 is weak in the atypical population. Therefore, the AHP is weak. And in some cases, and these are, again, back to the recordings, patch clamp recordings from single dopamine neurons in the VTA in anesthetized animals. So this is pretty cool, patch clamp in vivo, even if they are anesthetized. And Conoco found these bursts that they call plateau bursts because it's basically, it's like they're incomplete repolarization of the action potential, and then you get another one. And she represented the first one is in red. First one's in orange. Second spike is in red, and then a single spike is in black. So what you see is that single spike is short, less smashed in the phase plane. And so Johanna has, Tabea in the Roper lab has also seen spontaneously, but only in medial shell projecting neurons, she's seen these bursts occur spontaneously. And this is the burst, and this is in a slice. And this is the burst itself blown up. These are the phase plots. So this is the in vitro analog of those in vivo bursts. And Chris has modeled these. And what's shown here is just the doublets. But he's modeled this doublet, that you have the first spike and then it doesn't repolarize completely. And so you get another spike. And we, again, and this is the last slide. So we explain it in terms of phase plane again. So again, you can see the Z null climb that I've been talking about. It's kind of truncated here. And this is your trajectory of a single spike. It jumps at the knee from the quiescent branch. It spikes, it repolarizes, and it crosses the middle branch. That middle branch, once you cross that middle branch, it's the point of no return. You've got to now go to a quiescent state. And what happens with the plateau burst, we think, is that so this is the doublet, you're in this trajectory and the phase, and now this is the slow conductance, which is the SK plus the M currents, is now your slow variable. And as you repolarize, you don't make it to the middle branch. And so then you're free to produce another spike because you're on this upper spiking branch. And then finally, after the second one, you cross the middle branch and that stops it. All right, so I will stop here and just summarize the conclusions that the intrinsic dynamics of different dopamine neurons shape their differential responses to synaptic inputs. And Cowie and I are putting a lot of emphasis on this because you may be familiar with all these high-level models of dopamine action, maybe by Peter Diane or Randall O'Reilly, Michael Frank. None of these models treat neurons basically as like perceptrons. They don't treat them as real physiological entities. And so we'd like to think that their properties matter, and specifically that the properties of different subpopulations matter. And just to summarize, the atypical dopamine neurons have enhanced responses to excitation, and we suggest that this is due to lower expression of FGF12. Conventional dopamine neurons are more robust to synaptic perturbations, and we suggest this because of their deeper AHP, which recruits the A-type channel, and the slow variable is the slow inactivation that controls it. And then DLS projecting population has a unique response, so rebound response to inhibition, again, due to differences in the T and the S, well, the T and the KB4. And finally, the last point that I made very quickly was about plateau bursts. Since they're due to incomplete spike repolarization, that the atypical neurons that don't have a very strong AHP are much more susceptible to this type of plateau burst. So we think that the ones that Kaneko observed in the in vivo recordings were probably from atypical and not from conventional dopamine neurons. I know that was a lot, sorry.
Actually, I think I forgot to mention that Carmen is chair of the Department of Cell Biology and Anatomy at LSU, just because my brain doesn't work.
It's
that
okay.
much. But
I'm the only theoretical person. Everybody else in my department is either a teacher of anatomy or an electrophysiologist.
It's another unique thing, like a theoretician commanding biology.
What's a square peg in a round
I
hole?
figure she'd have the time when she doesn't have a wet lab to manage.
Yeah, right. But I'm also the oldest person in the department, so I attribute it to that.
Yeah, actually, I have two questions, and they are a bit related. This the part about the DLS differences in the brain neurons. So the first one, I think you kind of answer a bit, and I think that's part of the collaboration you have with Carmen would be: do you think that these differences in the way that they rebound are going to be behind some of the functional differences between DMS and DLS? And I see that you talk a bit about motion initiation. But there is also that difference between goal-directed versus habit formation. And do you have any insight about how the effects of...
It sounds like Cowie does.
Yeah.
Go ahead.
No, but
Mine's just a simple understanding. DMS cognitive, DLS habit. But I think if this rebound really is a movement initiation thing, that would be cool. Because Parkinson's patients have a terrible time with that.
Yeah. Sorry.
Oh, even in the real data, like the DLS rebound responses are very, very well correlated. Like they're all firing within like a 50 to 25 millisecond window.
But you're talking about your mom.
Yeah, but the DMS projecting cells have rebound ramps that vary from like 100 milliseconds to a second. And there's very little correlation in their rebound.
To be clear, these are all model predictions.
Well,
We believe them, but they're model.
I'm trying these data.
Oh, okay. So
in trying these data, like there's a very, like the first spike is very well constrained for the DLS, but
and I showed that the latencies. I showed that there was a slide that showed the latency.
Yeah, so the latency range for the DLS is like 25 to 75 milliseconds. The latency range for the DMS is like 100 to 2 seconds. So there's a wide range.
So there it is. That's also important.
So one important thing maybe for the audience to know is that those DLS neurons, they are the first ones to die of Parkinson's disease. They are by far the most accessible to death. And the fact that they are so unique, and it seems like it would be very hard to compensate for their loss of function, is definitely one of the things. So it's part of the R1 that we're writing.
Although we're submitting the NIDA, maybe we should submit
that was
NINDS someday.
So I'm already plugging this into my Parkinson's idea because that's part of the. So what would have to happen for a DMS neuron to become DLS neuron? Because they are a very, very unique population constrained by their expression.
Yeah, and that fibroblast growth factor 12 thing wouldn't do it.
Right,
that's not related.
That's the same as far as we can tell.
Yeah.
DLS.
And I saw a question in the back.
Yeah, just about the link to Parkinson's. So it's my understanding, I'm not an electrophysiologist, so I don't have to excuse me, but I thought in Parkinson's disease, the remaining dopaminergic neurons were more prone to burst firing. So do you think that they're trying to initiate movement, or is that?
I don't know. I always just thought they were trying to compensate. There's not enough dopamine, so they're upregulating to do more dopamine. But that's my simple-minded. Callie?
That's my Parkinson's Foundation back. Basically, just trying to figure if that's the case. If there's alternative ways that these other populations may try to recover and compensate. And so, for example, the idea of substituting one type of rebound burst by another type of burst
But have you considered in your model the loss of D2 auto receptors?
So we have well,
So
we looked at GERT. We looked at GERT.
channels in general.
Yeah.
And there's like a GERT tone that the cells receive, particularly in vivo, that modulates firing, and it does seem to make them more bursty.
Well,
Because-
it was really important.
Because it has some rectification to it, so once you get past the GERT, you're just free to have lots of excitation that the GERT.
Well,
can't
and the
really
GERT was
stop.
effective at depolarizing the dendrites,
Yes,
which recruits. Well,
hyperpolarizing the dendrites.
yeah. Sorry, yeah.
So these rebound experiments, they're applying current to the soma, but the T-type channels that are most interesting are distal. So you have some loss of the hyperpolarization, in that they're probably not as hyperpolarized as the soma is. But when you put on GERT, that basically biases it more hyperpolarized, and then we see a lot more T signals once the GERT is there.
And I really thought Nuri.
Yes,
I thought that your results were really interesting. That you have to go through the striatal circuit in order to use mind control to control a cursor.
I mean,
You had a second question.
yes,
Okay.
I did have a second question.
You have a good memory.
Yes. So this is more me, my complete ignorance about the extracellular part of, like you're recording, this is in vivo, and I try to understand, but is there any difference in the circuit between the DLS and DMS that could explain why these rebounds happen, the way they
Yes.
happen?
Well, so for one, we
Go ahead.
think that the cells that have a dendrite that projects, so
Oh, yeah.
the pars reticulata are exclusive to the DOS. So it wasn't actually like looked at like further projection, but it was looked at in terms of like the ADH1, I think it's the gene incumbent in negative or
I have
positive.
that in a slide somewhere.
But basically at least in terms of the genes expressed,
No, this
the
doesn't.
ones that have the dendrite and the SNR are consistent with the same genes that are expressed in the ones that project to the DLS.
Yeah. So then that would make it more.
So basically, we're
Yeah.
the only ones receiving a signal that's hyperpolarizing enough to substantially recruit T currents.
So
Because
only,
everything else.
Yeah, that's Rebecca Evans from
Yeah.
when she was in Zaid Colleague's lab.
so only these neurons that have this rebound burst that makes them rebound when they have a very particular type of hyperpolarization. They have a unique dendrite that goes straight into
The ventral, I stumbled over
Yeah.
that. Ventral
Yeah.
SNR is what I was looking for.
and they get like this gamma B input, the only gamma B input. So they can so that must be like super.
This is a good point. And Emily and I and Jochen and I, we're all hyper focused on intrinsic differences, but there's certainly differences in the circuit as well, and we can't ignore those.
Well, I would say this. I would say that the circuit and the intrinsic dynamics they're combined. Right, like I said, so
Well, we're arguing that the intrinsic dynamics are optimized for their role in the circuit.
Yeah.
that's I think in the R1, right? In
Right.
the text. But yeah, but think about
So which
this,
is what?
such a unique specialized thing with its own dendrite that received this input, and then they're the first ones to die. Like but yet so that's why the compensatory mechanisms, they must try to replicate these dynamics in some completely different way. Right?
But you
Speculation.
Yeah, speculation.
They do recruit other, besides dopamine, they recruit other neural modulators, right? The compensatory mechanisms.
They could. We don't know. That's all speculation. That's all.
I thought
no.
they were recruiting the cholinergic or.
That was Rebecca's idea, right? But that's
Which Rebecca?
Evans.
Oh.
Because we met her like just
Okay.
last week.
Oh, cool. I haven't seen her in a while.
Yeah. So I mean that's one thing. Maybe that they're doing this through a completely different neuromodulatory mechanism. But we do know that those DMS neurons in multiple models of Parkinson's, they fire more. And they reduce their KV4 function. So that also has implications for the shape of the faceplate, right? So that's also going to constrict.
What about the paper you just published with Laura?
Yeah, so that's one of those. Yeah, that's one of those. That's another discovery that we found. So you are still compressing the faceplate. So they're not going into the same type of mechanism of integration. So whatever happens must be. It's very likely a combination of circuits and intrinsic dynamic adaptations that are creating a different type of integration. Because I think one of the things and I think that's one of the very cool things about Carmen's work is that these simulations, they predict that these conventional neurons, they're doing a completely different type of computation with their inputs. Like they're probably doing something that like we see in electric fish neurons, right? Those
At least maybe at least some of the time, maybe.
yeah, like they're basically doing like spike timing processing, right? They're looking at it's also what happens in some auditory neurons, right? They're basically their signal is a difference in spike timing because they have such a regular pacemaking that's just going to be nudged by inputs. While the RPE like neurons, or projecting neurons, they are actually well designed to just follow their inputs, to excite when they're excited, to inhibit when they're inhibited. So it's a very fundamentally different type of computation. The only have in common really is they produce dopamine.
I look up another thing. The relating with the dopamine receptors, right? So you do have more prominent some D2 receptors in DLS.
Oh, that is. I forgot to mention that the cortical projecting ones don't have D2 receptors, right?
Okay. So would you imagine that these some of those changes may affect the way that the receptors are actually computing?
Yeah, I mean, there's a whole there's the what Nicholas is it Nicholas or Johannes that does the
That's the local communication. Yeah.
like a gradient, like some somatic dendritic release in some dopamine neurons affects others more than the other direction.
Mm.
But we haven't explored that, but
It does seem like the Gert tone within the NIBRA is directional. Like there's a hierarchy of who gets to be firing fastest, kind of in one.
I think Martin Vink may be looking at that too.
Yeah.
Let's see.
I think.
It's crazy. Yeah. So because you know that so another unique thing about dopamine neurons is that they also release some kind of dopamine from the dendrite. So they talk to each other with dopamine as well. And then but I don't know if beyond the cortical projection ones that don't express D2, if people have looked at if there is a more like if they're because it's not at all like the cortical ones don't have. But there might be also a gradient from conventional or non-conventional to conventional neurons in visual expression. And that basically tells who's the leader, who's the one who's listening to others versus telling the others what to do with their dopamine.
Sorry, the cortical neurons don't have D2 receptors.
Cortical
projecting.
Okay.
Sorry, cortical projecting. Cortical neurons do.
Project to
We can also have HTN to a degree,
yeah.
What?
despite otherwise being typical.
Oh, okay. Yeah, I don't
But you-
remember.
you
I don't
do
remember
kind
that
of
detail.
Kind of like, don't mention the cortical one when talking about atypical.
Okay.
Yeah. So
Yeah.
Atypical.
So the big thing is that as we, me being a psychology department, as we look into all these behavioral signals, the critical difference that goes completely ignored is that these neurons are very different. They're very, very, very different. I think Carmen's work is probably the one that goes deepest into the detail of characterizing this. And
Anytime.
yeah, we're hoping to now actually connect this, combine these cellular models with actual behavioral models, the circuit models, to and see how does this actually translate into behavioral signals?
That's actually why I'm here.
Yeah.
Right.
And then we can go into like what happens when one of them dies of Parkinson's and when they go right schizophrenia, then I think
Drug addiction.
Drug addiction. They're the ones give us money only.
True, we're funded by NIDA.
Yeah.
Why is that?
Yeah.
I can't remember. I made a connection somehow.
Yeah.
Okay.
Yeah.
Any additional questions? Does anyone actually right?
There might be a question on the Zoom because I think I saw a notification.
Ah,
And I'm incompetent to do that. Let's see.
I can
Chris,
almost died.
golly,
That's okay.
there's nothing there.
Chat. Yeah, there's a thing on the chat. No, just thank you for the talk.
Okay, cool.
Fantastic.
Yeah, I don't do more. I'm gonna log off. Right, well, anyway, thanks, Kelly.
No problem. So, any additional questions? If not, thanks again, Carmen.